I hope by now the classification criteria for the bacteria is quite clear in your mind. Now what we have to talk about is the point of difference between the gram positive and gram negative bacteria. We did discuss that the gram positive and gram negative bacteria, this consequence comes after when we have done the gram staining, and how the gram staining is done. What is the basis of classification? It is the peptidoglycan layer. Now we are going to see few points which you have to remember in terms of the distinguishing factors between the gram positive and gram negative bacteria. There are 6 of them mentioned here, and in the end I am going to tell you which one is more pathogenic and which one is responsible for causing most of the diseases in you and which one is not. Try to think which could be gram positive or gram negative. I will come to it in the end. If you can think by now, it's good. First of all, it is the staining. We did talk about this, is just repetition of the previous one. The gram positive bacteria, they retain violet stain, and the gram negative, they do not retain violet stain. This is the first one, and this is repetitive again. Why? Because the violet stain does not get controlled in the peptidoglycan layer. It becomes pinkish, these become pink, they retain violet. Talking about the outer membrane, gram positive bacteria do not have outer membrane, it is absent. In case of gram negative bacteria, the outer membrane is present. Now what is that outer membrane known as? That is known as glycocalyx. So these have glycocalyx, they do not have glycocalyx. Okay. Then we have, I would write here, glycocalyx, which could be present in the form of capsule, or it could be present in the form of slime layer. We are going to discuss that in detail. Then we come up to cell wall thickness. The next point of difference is which one is having a thicker cell wall. So certainly it is the gram positive bacteria which has around 20 to 80 nanometer thick cell wall. They have a thinner cell wall, but it has layers, and the cell wall thickness varies from 8 to 12 nanometer. See the difference. There is a lot of difference between their cell wall thickness. Talking about cell wall, how is the cell wall? This was about thickness. How is the cell wall? They have a smooth cell wall, completely made up of peptidoglycan. Again the term comes. These gram negative bacteria, they have wavy cell walls which could be joined to plasmolema at certain points. Okay, so the cell wall is wavy, and it would be such at this would be plasmolema supposedly. So the cell wall would be joined at certain points, such points, that would be the locus. So the wavy cell wall is present. Then we talk about murin content. Murin is a complex carbohydrate again. It is around 70 to 80% in the gram positive bacteria, while in gram negative bacteria it is less than 30%, it is 10 to 20%. So this is another point of difference. Then comes teichoic acid, how much it is present, and if at all it is present. Teichoic acid is present in the case of gram positive bacteria, while in the case of gram negative bacteria it is absent. So these are the points of differences between gram positive and gram negative. Now coming to the question in the beginning, few of the gram positive bacteria are pathogenic, while most of the gram negative bacteria are pathogenic, they are disease causing. So this particular class of bacteria is highly notorious. So we add pathogenic species, many pathogenic species, okay, and these are pathogenic. Few of them could be pathogenic, but not all of them. So few are pathogenic. While they are mostly pathogenic. So they are more notorious. The notorious ones are gram negative, N for notorious, N for negative. You can remember it that way, and these are the points of difference that you need to remember.